Hi everyone, our drug is azithromycin.
And this was our team. I'm Sarah Gonzalez. My major is animal science, and career interests include wanting to be a wildlife vet and work in animal rehabilitation, or also on the other side of the spectrum, I might want to be doing small animal vet and work in small shelters.
My name is Mania Mohebi, and I'm a biology major and a pre-med, and I hope to be an anesthesiologist.
My name is John Abdallah. My major is biology, and I'm hoping to be a dentist in the future.
My name is Aya. My major is in bio. I'm also a pre-med and I hope to be a cardiologist.
My name is Ani. My major is also biology, and for my career interest, I hope to be a dermatologist.
My name is Param Gulashani. My major is anthropology, and I want to be a doctor.
Here we have the 2D structure. As you can see on the left, all the hydrogens are shown, and that's the actual structure. But on the right side, we excluded all the hydrogens just to make it look simpler. Here we have the chemical formula, which is C38H72N2O12, and we have a 15-membered azolide ring, as you can see right here, and 2 sugars. Prem, can you tell us what an azolide ring is?
An azolide ring is a macrolactone ring containing nitrogen.
What is a macrolactone?
A macrolactone is an 8 or more membered ring containing cyclic esters. An example of a cyclic ester would be right here, where you can see the carbonyl carbon, that alpha carbon, and the ester oxygen. Also, we found out that azithromycin is an amorphous solid, which basically means the atoms or molecules are not in a definite or fixed lattice pattern. Common examples of this would be glass or plastic.
Some physical properties of this molecule are that this molecule is like a white powder and it is not very fine.
As you can see, this is the 3D structure for azithromycin. Notice the 15-membered azolide ring along with its 2 unique sugars. Also, if you look closely enough, you're able to see the cyclic ester. I'll just give you guys a moment to look at it.
For the functional groups, the most important one to identify was the lactone group, which is an ester within a ring. This is because it is very important to the function of the molecule, and it also gives it its identification as a macrolide. And a macrolide is a series of natural products that have a lactone group bonded to 1 or more deoxysugars. Here we can see azithromycin, and we can see the lactone group, which is an ester within a ring, labeled in red. And we can also see the deoxysugars, one right here and one right here. This is another example of a macrolide, which this is erythromycin. And we can also see the lactone group labeled in red and 2 deoxysugars labeled in blue. For other functional groups, we found 2 tertiary amine groups, one right here and one right here. We found 5 alcohol groups labeled in yellow, 1, 2, 3, 4, 5. We also found 1 ether group, which is connected to the deoxysugar, as well as 2 acetyl groups, which are OCO groups, as seen here. This was tricky to find in the beginning because it looked like 2 ether groups, but it is actually 1 acetyl group. Here we see again the ester group, which is within the big lactone ring or lactone group. For intermolecular forces, every functional group identified can do hydrogen bonding and have dipole-dipole forces. They all have dipole-dipole forces because they have electronegative atoms, such as nitrogen and oxygen. They can also do hydrogen bonding because they either have an H on the electronegative atom or they have a lone pair, which can act as a donor for another molecule, such as water, which will act as an acceptor for the electron pair. Just note that these amines are tertiary, so even if they form hydrogen bonds, they will be weak because they are very bulky, and that will lead to lower boiling points.
Here we have erythromycin and azithromycin. In my research, I read that erythromycin is a common starting point in order to form azithromycin. That is why I chose to do the synthesis starting from erythromycin. In order to start the synthesis, I had to see what was different between erythromycin and azithromycin. I noticed that erythromycin has a 14-member ring and a ketone, whereas azithromycin has a 15-member ring and an amine group. So how do we make a ketone into an amine group, and how do we expand the ring? So in order to turn the ketone into an amine, we need to do it in the steps of ketone to oxime to amine. So in order to turn the ketone into an oxime, we need a weak base and a buffer, in this case hydroxylamine. The N attaches the carbonyl carbon and causes the double bond here to become a lone pair on the oxygen. This makes two different bonds onto the carbon, one with the oxygen and one with the H2NOH. Then there's a proton transfer between one of the H's on the hydroxylamine and the oxygen's lone pair. This proton transfer creates an OH. The OH needs to leave, but since OH is a strong base, which makes a poor leaving group, it can't. But it manages to leave because the nitrogen's lone pairs displace the OH in an elimination reaction. The OH leaves and the lone pair of nitrogen forms a double bond here. This now creates an oxime group. The oxime needs to become an amine, and so we remove the H's by using a base, maybe OH. The base binds with the H on the nitrogen and the H on the oxygen. That leaves us with a carbon double bond nitrogen to an oxygen. In order for the oxygen to leave, it is reacted with TSCl and becomes TSCO, and that's a really strong leaving group, so it's able to leave. Additionally, in order for the ring to expand from 14 to 15 member ring, we take the 15th bond and bond it to nitrogen so that instead of a 15 to 2 bond, it is a 15 to 1 bond since nitrogen is now 1. There's also an intermolecular SN1 reaction with OH here. It binds with the carbon 2, and this is necessary because in the later reaction, this bond will be broken and will become a source of hydrogen for the nitrogen, which is needed for a later reaction. Now, the ring is expanded, and there must be a reduction in order to remove the oxygen from the structure. Reductions are commonly assisted by sodium borohydride. The BH4, which is a hybrid nucleophile, attacks at the carbon double bond N, amine, and breaks the double bond, and an H attaches to the N. Also, the H from the CH3OH is positive since the oxygen is partially negative. And so the negative O reaches for the positive H and forms an OH again over here. Lastly, the structure goes through a reductive amination, which is when the oxygen from formaldehyde bonds with the H of nitrogen, and then the CH2 part of formaldehyde bonds with nitrogen. This creates an amine, which under acidic conditions will be protonated and form an iminium ion, which is later reduced using NaBH3, which is similar to reducing agent NaBH4. The reduction will break the double bond between nitrogen and carbon and add an H to nitrogen. Essentially, this mechanism installs a methyl group to the nitrogen. And that's how you get azithromycin.
Now, the function of azithromycin as a drug relies on its structure. This drug is a macrolide antibiotic with a 15-membered lactone ring, excellent at treating bacteria and inflammatory diseases. Macrolides work by binding to a specific subunit of ribosomes in bacteria, which inhibits the formation of its proteins, thus stopping bacterial cell growth and spread. Specifically, azithromycin inhibits bacterial protein synthesis by binding to the 50S ribosomal subunit of the bacterial 70S ribosome. This binding inhibits peptidyl-transferase activity and interferes with amino acid translocation during the process of translation. This is the process that forms peptide bonds between two adjacent amino acids in the synthesis of the bacteria proteins. So if we look at this diagram, we can see the mechanism in which macrolides like azithromycin work. Bacteria are usually 70S subunits made up of a 50S and 30S ribosomal subunit. Azithromycin, which is represented here by macrolides in red, bind reversibly to the 50S subunit of the bacteria over here and stop the peptidyl-transferase step of protein synthesis. This just means that this peptide chain, which we see here on the A site, can no longer transfer to the P site, and this stops the chain's elongation right then and there. Basically, by binding to these ribosomes, the drug prevents the growth of bacteria by stopping its synthesis. In regards to COVID-19, macrolides have been known to increase the pH of the Golgi network, And this alters glycosylation, which is a process that is necessary for enzyme receptors, specifically the angiotensin converting enzyme 2 receptor, also known as the ACE2 receptor. This is a cell surface enzyme receptor to which COVID-19 is believed to bind, which we have represented over here. By inhibiting glycosylation of the receptor, we can inhibit the virus from binding to the host cells. Research done by the University of Oxford has also seen significant reduction in the levels of an enzyme called furin. COVID-19 is believed to possess a furin-like cleavage site in the spike protein, which we see here, that facilitates the viral entry into the host cells. A study conducted at the Infection Biology Unit of the German Primate Center determined that furin allows the virus, specifically through the spike protein, to fuse to the ACE2 enzyme receptor, which allows the virus to deliver its genome into the cell, further replicating within the host. So it's possible that azithromycin, coupled up with certain malaria drugs, can prevent this viral entry and infection into host cells.
So first, let's start off with some background information about azithromycin. It is currently used as an antibiotic to treat respiratory, skin, eye, and ear infections, as well as some sexually transmitted diseases. Some of the most common illnesses it is used for are bronchitis and pneumonia. As for if it's naturally occurring, azithromycin is a modified version of erythromycin, which is a naturally occurring macrolide derived from Streptomyces erythris. This would answer the next question of whether it is synthetic. Azithromycin is a semi-synthetic antibiotic. This means that the molecule comes from a natural antibiotic that has been chemically modified in the laboratory to enhance stability. Essentially, it is a derivative of a natural antibiotic with slightly different but advantageous characteristics. As for its discovery, it was discovered by scientists working for a Croatian pharmaceutical company by the name of Pliva in 1980. The team of scientists who were credited with the discovery were led by Dr. Slobodan Dokic. Pfizer currently commercializes azithromycin in the United States, and the tablets are manufactured at the US FDA certified formulation plant in India. Where our molecule is with respect to drug delivery is that it is on the World Health Organization's list of essential medicines, and is considered to be the safest and most effective medicines needed in the health system, and is classified as critically important for human medicine. It was FDA approved in 1988 and it is currently sold under the names Zithromax and Zmax. Because it is FDA approved, it is ideal to consider for treatment for the coronavirus because developing a new drug and seeing it in the marketplace could take up to 10 years, not to mention research and development would cost roughly about $2.4 billion. So the bigger picture here is in how azithromycin relates to COVID-19. Since it is an antibiotic on its own, it is ineffective against viruses. However, when adding it to chloroquine or hydroxychloroquine, which are both drugs designed to combat malaria, there's been limited success in the treatment for the coronavirus. The initial data about azithromycin came from an observational open-label study done in France. Unfortunately, open-label studies in which the doctor and patient are aware of the treatment makes it difficult to isolate variables and say whether a particular drug is working. In this study, French doctors were testing chloroquine and hydroxychloroquine on coronavirus patients. And it turned out 6 patients among 20 that happened to be taking azithromycin for other conditions all recovered. This excited researchers, but after several experts worldwide analyzed the study, there was a consensus that the study showed several methodological limitations, one of them being the lack of randomization of the experiment. They concluded it did not provide useful evidence to make any valid or trustworthy conclusions. However, even though there were positive results for that study, there have also been negative ones. For example, in another study done with U.S. veterans that is currently awaiting peer review, patients treated with hydroxychloroquine alone experienced a death rate of 27.8%, while patients treated with both hydroxychloroquine and azithromycin had a death rate of 22.1%. But patients receiving neither treatment had the lowest death rate of 11.4%, as you can see in the chart. This showed that hydroxychloroquine and azithromycin had no positive effect, and hydroxychloroquine alone even had a higher death rate. There was also no benefit in ventilation rates either. The National Institutes of Health is now specifically recommending against the combination of hydroxychloroquine and azithromycin for patients suffering from this virus, because there's just not enough evidence. These findings highlight the importance of waiting for the results to have solid confirmation before the adoption of these drugs. Some hydroxychloroquine proponents have said that the trials finding no benefit are flawed and that the drug is given too late, and others say the cause of the failure for hydroxychloroquine treatment should be investigated by testing the isolated coronavirus strains of the non-respondents and analyzing their genome to see why it's working for some and not for others. But overall, there's just some positive proponent to using azithromycin, but there's just not enough evidence and it hasn't been studied in depth, so it can't be used as a treatment option. And here's our works cited. And this concludes our presentation. Thank you for listening.